So we're moving on to leukocytes, and there are various components of our leukogram that we're going to talk about. But why are leukocytes called leukocytes, they're also called white blood cells or WBCs, is in part, or possibly all of this, is because of their color. So if you spin down blood, kind of like when you did in lab looking at your PCV, you have your red cell component and your plasma component, and then that white line in between, those are your white blood cells and your platelets, and that's of course called your buffy coat. And in an animal who would have a marked leukocytosis or even leukemia, who could have hundreds of thousands of white blood cells, you could make a very, very thick buffy coat, but usually it's around 1% normally. So when we talked about our red blood cells, those were in the millions, of course. Millions per microliter, at least usually 5 million, whereas our white blood cells are going to be in the thousands normally per microliter, and that's your actual total number of leukocytes. So how you actually get a total number of leukocytes is a hematology analyzer. And so although you can estimate it on the slide, it's by far more accurate if you have a good analyzer that can give you your total white blood cell count. So that's kind of our first measure of white blood cells, is your total white blood cell count. So that total white blood cell count that you get from your analyzer is per microliter. And again, we practiced very briefly in lab estimating white cell counts, or maybe you did. But what is more meaningful after your total white blood cell count are your absolute counts. So your absolute counts of your various leukocyte types. So this absolute count of your leukocyte types is really how we interpret a leukogram. And the other part that we'll talk about is leukocyte morphology. So we'll actually look at what our leukocytes look like and our various types to tell us things such as inflammation or neoplasia when we look at our white blood cells. So let's refresh our memories on the different types of white blood cells that there are. So a hematology analyzer can give you a leukocyte differential. And a differential is essentially determining from however many cells it counts the percentage of each cell, of each leukocyte anyway. And we're going to use white blood cell and leukocyte interchangeably. And this can be automated. And I'm going to put a frowny face next to this, because most of the time analyzers do a very poor job of an automated differential, with the exception of some standard species and very normal animals. So most of the time we do a manual differential based on the various leukocyte types, and then we take that percentage for each leukocyte type times our total white blood cell count, and that's where we get our absolute numbers. And so the various types of leukocytes that we enumerate in most species, not avian species and not reptiles. So we have what we call a segmented neutrophil, and that's our most numerous in dogs, horses, and cats. We have a band neutrophil, which is one before the segmented neutrophil, and it's a little bit immature. So these are called granulocytes, and we'll talk more about why they're called that. Eosinophils are relatively uncommon, and depending on the species, the pink or red granules look a little bit different. Basophils are very rare, and they have kind of purple granules, and we'll talk about why that is. In birds and in reptiles, they have heterophils, and we call in guinea pigs as well, pseudoheterophils, as well as in rabbits. I'm sure you found the frustration that monocytes are in lab. They look like everybody, and monocytes are pretty infrequent. And then lymphocytes are kind of the number one leukocyte in ruminants and in many rodents. And so when you do a differential, you count each of these, and you get percentages. So you might get your percentage of neutrophils, lymphocytes, monocytes, and you may have eos, and rarely basos. So you get these percentages, and your total should be 100%, and so you have counted 100 cells, and then you multiply that times your white blood cell count, and that's per microliter, and then that gives you whatever the number is. So you take that percentage times the white cell count, and then you get whatever your neutrophil count is per microliter. And these are the numbers that you get on a CBC, and we will practice this a little bit. So look at this image for a second and see if you can identify the various leukocyte types. I'll give you a second. And then also tell me what that cell is right there. And we'll regroup in a second after you've looked at these, and then you can hit pause and figure these out, and then I'll tell you the answers. All right, so these cells are all my neutrophils here. All these cells are my neutrophils. These 3 cells are my lymphocytes, and here is an eosinophil, and there's no monocyte in this field. So if my analyzer gave me a total white blood cell count of 10,000 cells per microliter, I've just done a manual differential, and let's say that there were 100 cells we counted, so in fact there were only 10, and so in that case 60% of them were my neutrophils, 30% of them were my lymphocytes, and 10% were my eosinophils. I would take the percentage and I would multiply it times my total white blood cell count. So that would be 0.6 times 10,000, which is 6,000. And of course, 30% times 10,000 is 3,000. And 10% times 10,000 is 1,000. And so those would be my absolute counts, and then I would compare them to my reference range. So one thing to note is that so this cell right here is a nucleated red blood cell, also known as a metarubricite. And depending on the kind of analyzer you have, it may erroneously count these cells as leukocytes because, of course, there's a nucleus, and some what are called impedance analyzers or technology that uses impedance where it's only looking for sort of a interruption in light flow would count this as a leukocyte. So if you have greater than 5 NRBCs per 100 white cells, because that's how we enumerate them, you would have to do something called correcting the white cell count, and we did that in class once, and I'll show you quickly how to do it. I think we've got one more time that we'll do it during class, but you really need to know what kind of analyzer you have if you're going to do it. Also, if you're estimating on low magnification how many leukocytes you have, you want to make sure you don't have a lot of nucleated red blood cells because, of course, they look a lot like lymphocytes, and so you don't want to count them as lymphocytes because an animal with a regenerative anemia might have a lot of these hitchhiker nucleated reds, and you might mistakenly think that it's a lymphocytosis. So how you do a corrected white blood cell count is you count your nucleated red blood cells per your 100 white cells, and you take that number and you put it into this equation here. So that's your leukocyte fraction. And then you take your total white cell count and multiply it by that. So if our white cell count impedance, meaning that the analyzer couldn't tell the difference, that would be 30,000 times that's 0.83. And that would equal 24,900 per microliter. And that would be your actual corrected white blood cell count. And then you would use that to do your differential count. So you would take that times your various fractions.